investigations of palm oil derivative beta-carotene in diabetic vascular dementia in rat. This work is carried with my co-researchers, that is Mr. Lim K. and Dr. Varadarajan. And this work is carried out in the AIMS University, Malaysia. Research background is that vascular dementia is the second most common causes of dementia, and it is one of the next level of the vascular dementia disorders after Alzheimer's disease. And this both the disorder is coming under the based on cerebrovascular dysfunction, and various risk factor is employed in this vascular dementia development. And diabetes mellitus, particularly type 1-2 diabetes mellitus, is one of the major risk factor for vascular dementia. And however, no medication is approved for this specific management of this vascular dementia. Certain natural compound is tested in the clinical level as well as preclinical level is tested for prevention of vascular dementia progression. And a certain report is evidence that carotenoids is beneficial for cognitive performance. And this carotenoid compound is highly rich in the palm oil product, and palm oil is one of the richest plant and source from the Malaysian community, or Malaysia is producing large palm oil product and palm oil industry. Next, problem statement and hypothesis. And a major problem is that specific treatment medicine is not available in the clinical setup. So that multiple drug development or drug discovery is essential for the prevention of vascular dementia disorders. And hypothesis based on this, that we are found that we are trying to find out the role of beta-carotene in the management of neurovascular complication and neurological dysfunction, and particularly in vascular dementia conditions. This is the research hypothesis flowchart, and intravenous administration of streptozotocin will cause the diabetic mellitus. It is well-known fact in the experimental animal model, and however, it is producing a free radical pathway activation, and acetylcholinesterase pathway is activation, and destroy the endothelial cells and acroglial cells in neuron system. So together, it is accelerate the endogenous driven system is declining, neurodegeneration is accelerating, metabolic toxin is accumulating, and nitrous oxidative stress it is improving, elevating, and increase the vascular unit dysfunction and neuronal death, and finally it is also showing the diabetic vascular dementia. But what is the role of beta-carotene? Beta-carotene is well-known fact to prevent the free radical due to antioxidant property. But that reaction on the acetylcholinesterase pathway and microglial endothelial cells pathway and other regulations of diabetic vascular dementia conditions, it is not explored yet. This is the aim and objectives. And the first aim is to investigate the role of beta-carotene action in experimental model of vascular dementia. Second objective is to identify the pathophysiological mechanism of vascular dementia progressiveness. Third one is to explore the possible regulatory mechanism of beta-carotene in the management of diabetic mellitus associated vascular dementia. So we are made experimental designs. Male sparkle rat were used in these studies. Total 5 groups we are used, and each group consists of 12 animals. And group 1 served as a normal control. It is without administration of any drugs. And group 2 served as a streptozotocin 50 mg per kg intraperitoneal administration for induction of diabetic vascular dementia. So remaining 3 groups also same methodology is added, and additional that group 3 is added administered beta-carotene 50 mg per kg orally for 15 consecutive days once the diabetic is confirmed. And group 4 also beta-carotene 100 mg per kg orally administered for 15 consecutive days. And group 5 it is vascular dementia plus donepezil 1 mg per kg administered orally for 15 consecutive days. So this is the methodology flow chart. It is nicotinamide and streptozotocin 50 mg per kg intraperitoneally administered, and so that it is able to develop this type 2 diabetes mellitus. And day 3 onwards, and day 3 and day 7, it is estimated the blood glucose level to confirm the diabetes mellitus. And day 8 onwards, this drug administration is started, that is beta-carotene and donepezil drugs. Donepezil here employed as a reference control drug. So day 8 to day 27 is drug administered once a day, and day 18 to 21 is Morris-Waterman test, that is the memory, learning and memory test. Learning function is assessed the first 4 days, and memory function assessed in the day 22, that is Morris-Waterman test. So first 4 days means it is day 18 is considered as a day 1 for Morris-Waterman test. Okay, and day 23 it is animal sacrification and sample collection is made for biochemical estimation. This is the Morris-Waterman learning and memory behavior assessment test device. This is called Morris-Waterman pool tank, and this is established method. It is circular pool, and it is divided into 4 quadrants, each quadrant named as a Q1, Q2, Q3, Q4 quadrant. And this Q4 quadrant is a target quadrant. It is here platform is kept for first 4 days assessment. So all animal is exposed in the all quadrant in every day, so that that animal is completely understand the platform location. Once the platform location is reached, then it is considered as the animal is land. So that is termed as escape latency time. It is first 4 days we are assessed. And in 5th day, platform is removed and animal is placed. Animal already learned, so that it is able to retain their memory function is there. Then we can consider as a time spent in target quadrant level will be higher. So if vascular dementia animal not able to spend more time in this target quadrant, this is the basic logic to assess the learning memory function. And the end of the day, that the animal is sacrificed and blood sample collected, and brain tissue also isolated immediately and homogenated with phosphate buffer. pH 7.4 is maintained. The supernatant of brain homogenate is collected with the centrifugation process, that is 3500 rpm for 15 minutes, and this sample is stored in the -20 degrees centigrade for further biochemical estimation. Biochemical estimation, total protein is assessed by low resettle method, and spectrum automatically it is detected in that 750 nanometer wavelength, changes of color absorptions, and a standard plot was made with 0.2 to 2.4 milligram ml of point serum albumin as a reference standard. Brain acetylcholinesterase activity also assessed with the method of Elman et al. 1961, and here changes of absorption is measured at 412 nanometer wavelength. Here that formula based that acetylcholinesterase estimation is made. And here result section, this effect of beta-carotene on ELT in Morris-Waterman test. Normal rats have the significant learning ability, that is indicating the decrease the escape latency time on day 21 when compared to day 18. And here you can see the flow pictures. This is the significant improvement. And vascular dementia showed the significant impairment of learning ability compared to control group, that is the red color box. And administrations of beta-carotene shown the ameliorative effect in the diabetic vascular condition, that is green color, and blue color is indicating reference control drug. It is almost similar to the beta-carotene action is similar to the donepezil drug action. Similar result observed in the time spent in target quadrant, that is assessed in the end of the experimental protocol, that is assess the memory function by using Morris-Waterman test. So here you can see normal animal Q1, Q2, Q3, Q4. That Q4 level is elevated, and in vascular dementia condition that Q4 level is declined. So it means that animal is in memory impairment is there. So after treatment of beta-carotene and donepezil, it is significantly improved the time spent in target quadrant. It is indicating the drug treatment is showing the significant memory improving action. Here, that another one is biochemical estimation, effect of beta-carotene on acetylcholinesterase activity. So in diabetic vascular conditions, acetylcholine level is elevated, and we are assessed in the various parts of the brain tissue, that is hippocampus, cerebellum, extracranial cortex, amygdala, septum, because these are the areas is also implying the memory functions in my major to minor. Next one is treatment of beta-carotene. It is and donepezil. It is significantly decreased acetylcholinesterase activity, so that the acetylcholine level remain is higher, so that it is able to producing cognitive function. So beta-carotene shown effective in the donepezil regulatory actions in acetylcholinesterase activity. So this is the result is showing in this tabular form that diabetic vascular dementia condition, that is acetylcholinesterase level is increased. But the beta carotene and tonopacil, it is improved that acetylcholinesterase activity. Next on is discussion. So morisotamide result to the vascular dementia group had longer ELT and shorter time span in target quadrant. It is indicate the improvement of better memory from in beta carotene is improved the this memory function via regulations of ELT and time spent in target quadrant. And this is also evidenced by that 2019 studies. Beta carotene is improved the performance of obstructive sleep apnea syndrome conditions, and it is indicating the sort of the ELT level, but mechanism is inhibition of expression of caspase-3 and tau protein expression is reduced. Next, other carotenoids, that is particularly astaxanthin, also show the beneficial effect in the terms of memory. It is tested in the status epilepticus conditions of animal, and here showing the long time spent in target quadrant and inhibit the neuroinflammation and hippocampal damage. In further, crocin is also one of the carotenoid member. It is also showing the shorten of escape latency time. This better spatial memory function is showing in this experimental animal studies. And so acetylcholinesterase is playing a vital role in the regulations of cognitive function, and because it is improve the acetylcholine level in the brain system. So Hira et al. 2019, he suggested that administrations of beta-carotene is increase the acetylcholinesterase activity. And in addition, in silico talking studies also, beta-carotene potentially binding to the acetylcholinesterase active enzyme protein, and it is regulate the acetylcholine level. So in addition, beta-carotene lowest concentration, 300 to 400 micromole, it is also inhibit the acetylcholinesterase activity. So it similar result is observed in our results, current results work, and beta-carotene attenuate the elevated acetylcholinesterase activity in diabetic condition. So based on these aligned findings, previously the hypothesis administration intraperitoneal administrations of streptozotocin causing the diabetic vascular dementia. So beta-carotene play a role in the acetylcholinesterase and this regulation of diabetic vascular dementia. This is our hypothesis or research question. So this hypothesis and research question is proved. Beta-carotene, yes, it is, of course, it is regulating the acetylcholinesterase activity, free radical regulating function, and also it is improved the diabetic vascular dementia condition from the disease state to normal state. So in conclusion, beta-carotene has shown to be beneficial in vascular dementia via inhibiting the acetylcholinesterase activity. It is particularly improving the spatial memory function, and so it is can be act as a newer natural compound for the treatment of vascular dementia condition. So potential beta-carotene ameliorating the neurodegenerative disease also can be tested, and further research work can be conducted in the clinical setup level due to the based on this result. So neurodegenerative disorders in very common in the clinical setup. So beta-carotene can be tested for Alzheimer's disease, Parkinson's disease, including vascular dementia condition, because vascular dementia is one of the neurodegenerative or neurovascular disorders. These are the references we used in this research work. And acknowledgement, we are grateful to Malaysian Ministry of Education for financing this research work, and via Fundamental Research Grant Scheme. This is the number. It is approved by 2019 by Malaysian government.